they contributed their works in finding these enzymes. So enzymes, they are the catalyst which promotes or which helps in promoting biochemical reactions in living organisms. And these enzymes okay, boys, okay, students, these enzymes, basically they are made with proteins, and there is a statement. According to that statement, all the enzymes are made with proteins, but all the proteins are not enzymes. So basically, enzymes are made with different types of proteins, and there are nearly 64 types of proteins are there, and right. These enzymes, they are made with proteins, and out of 64 proteins, few of them participates or few of them uh, are used in the synthesis of enzymes. And Louis Pasteur stated that ferments a word, and this ferments the word, it is connected to microbes. These microbes, they helps in fermentation process, and this fermentation process was proved by taking grape juice, and through this process it was changed into alcohol. And actually, originally this enzyme term, it was named by W. Punney, and term zymase was coined by Edward Buchner. Then James Sumner, scientist, he isolated urease enzyme from jack bean, and postulated that all enzymes are proteins. Just only we said that all enzymes are made with proteins. So this statement was given by doing after this experiment, and that was done by James Sumner. He isolated urease enzyme from jack beans and postulated that uh, all enzymes are proteins. Then some nucleic acids that have like the enzyme 23S, rRNA, ribosome, and prokaryotes. And 28S rRNA is found in eukaryotes. And if we observe the structure of enzyme, if we observe the structure of enzyme that appears in this manner, that appears in this way, in which we are observing some pink spots which are called as active sites. So these active sites are the centers for reaction. Okay, so substrate molecules, they comes and fit in this active sites, and both together makes a bond between this enzyme and the substrate molecules. They frames a bond, and they undergoes reaction, after getting end products, those end products will come out from the active site and enzyme remains as it is made. So this is the structure of enzyme. On its surface, it is having a lot of active sites where the substrate molecules comes and fit, and a bond is laid between this substrate molecule and active sites. And after undergoing reaction, this end product and enzymes both will be separated from one another, and enzyme comes out as it easily, and end products comes out from the active site. So this is about the structure of enzyme. Here, just only we said that enzymes are made with proteins. And in proteins, again, we have different types of proteins. And these enzymes, especially, they are constructed by tertiary proteins. And here is the diagram of tertiary protein structure. We can observe this protein, tertiary type of protein in which, say, one amino group is there, and this tertiary protein, it helps in giving the shape of the active sites on the enzyme. Then, if we observe the chemical reaction, this chemical compounds, they changes, uh, chemical changes, chemical compound changes. There are two types. One is physical changes and the other one is chemical changes. And the bonds are broken and new bonds are formed during the transformation is called as a chemical reaction. For example, barium hydroxide, which combines with H2SO4, results in the formation of barium sulfate and H2O. And rate of a physical or chemical process refers to the amount of product formed per unit time. So that is shown in the form of a equation. So here, what we are understanding is chemical reactions, there are two types. One is physical changes and the other one is chemical changes. And in both the reactions, the simple formula is bonds are broken and new bonds are formed during the transformation from substrate to the end products. And it is called as a chemical reaction. Then catalyzed reaction. Another type of reaction is catalyzed reaction. And here is the example. What is that? CO2 plus H2O gives rise H2CO3. And in this, metabolic pathway is also there, in which glucose molecule that will be converted into two pyruvic acid molecules. And this process is called as glycolysis. And this glycolysis, it includes various types of enzymes in getting two pyruvic acid molecules from a, single glucose molecule. And that is shown in the form of a equation that is C6H12O6. It is a molecular formula of glucose, and when it combines with oxygen, and this oxygen has a nature of burning. Oxygen that burns or that undergoes aerobic respiration, in which this glucose molecule, six carbon molecule, that will be converted or changed into uh, two molecules of 3C compound, which is called as pyruvic acid. Along with this pyruvic acid, two water molecules also be released through this process. So in this also we can understand uh, this respiration is a metabolic pathway. It is one. It is a part of metabolism, and in this metabolic pathway, respiration takes place, and this respiration includes two types: aerobic respiration and anaerobic respiration. And this aerobic respiration equation is CO2 plus H2O. Sorry, aerobic respiration. In aerobic respiration, glucose molecule will be combusted, or glucose molecule is burnt, and finally it releases two pyruvic acid molecules along with the water molecule. And this is the first step in aerobic respiration, which is called as glycolysis. Then, in this glycolysis, each and every step, it involves enzyme. Under the influence of enzyme only, this glucose will be converted into two pyruvic acid molecules. So this is the importance of enzymes in metabolic pathways. Right. This is the uh, chemical conversion. That means enzyme action. This diagram indicates a enzyme action. Here is a this yellow color is a enzyme, and all this green portion is active site. So here this enzyme having with active sites. Then these are the substrate molecules. Here substrate molecule shape of the substrate molecule and a shape of the active site. Both are similar or the resemblance in their shape. So substrate molecules with the same shape of the active sites, they comes and binds with the active site, or first substrate molecule that comes and sit in this active site. Then a bond is laid between this active site and substrate molecule. See the next diagram. Here it is, this is a substrate molecule and this one is a enzyme. This active site that binds with the substrate molecule in giving enzyme substrate complex. See here it is a complex enzyme with substrate molecule results in the formation of a complex. Now reaction takes place between these two, and finally here substrate will be converted into product, and this product will be released from the active site that comes out, and the enzyme remains as it is without undergoing any change. It remains as it is. So as we said in the first slide, that enzymes are catalysts. They participate in the reaction without undergoing any change. That gives the end products. So enzymes are treated as catalysts, and enzymes also be called as biocatalysts. Why? Means these enzymes are extracted from living microbes. That's why they are also be called as biocatalysts. Here is a graph, and the graph is laid between potential energy or activation energy and progress of reaction or rate of reaction. Between these two issues, a graph is laid. Here, substrate molecules which participates in reactions will have some energy. Initially, they will have some energy which is called as activation energy or potential energy. And with that starting potential energy, substrate molecules, they participate in the reaction. And if we observe the progress of reaction, that gradually increases, the rate of reaction will be gradually uh, raised or increased by utilizing this potential energy or activation energy. And by using this potential energy, they reaches one point which is called as transition state. At this point, or to reach this transition state without enzyme, the substrate molecules requires a lot of potential energy or activation energy. That means without enzyme participation, a reaction requires a high rate of potential energy or activation energy. Then after reaching this transition point, gradually there at this juncture or at this point, transition state point, substrate molecules, they combines with the or they binds with the sorry activation sites or this at this transition state, substrate molecules, they make the bonds with the active sites on the enzymes, and a temporary bond is laid between the substrate molecule and active site. As a result of the temporary bond, a complex will be developed, and that complex is called as enzyme substrate complex. And that complex is laid or developed during this transition state. After reaching this transition state, gradually a bond is diluted and end products are produced. This end product molecules, they comes out, and enzyme as it is it comes out. So here, without the participation of enzyme, this transition state is developed by utilizing high level of potential energy or activation energy. After that, gradually end products are released. And now, if we observe with the participation of enzyme, with the participation of enzyme, is the transition state reaches easily or toughly? That will be observed in this graph. Yes, this substrate molecules by using this activation energy and with the support of enzyme, they reaches the transition point very easily by using less amount of activation energy. Why? Because here enzyme is helping in giving the end products. So we can easily understand this. Without enzyme, transition state is developed by using a lot of potential energy, but with the participation of energy as well enzymes, this substrate molecules, they reaches the transition state with a little amount of activation energy only, and that gives end product molecules. So by this graph, we can understand that transition state with uh, potential energy uh, is developed by using a lot of activation energy and without the participation of enzymes. And here, this blue color graph parabola, it indicates what? Means with the support of enzymes, a transition state is created or developed with the enzyme support. Less amount of activation energy is used in giving end products. So this is the concept of activation energy. So finally, totally what we can understand is activation energy is every substrate molecule have activation energy, but this activation energy sometimes it is used more in the absence of enzyme, and activation a less amount of activation energy is used with the support of enzyme. That we can understand by this graph. Then explanation about the graph. If P is lower level than the S, P means potential energy. Its level is lower level than substrate level. It is an exothermic reaction. And one need to supply it energy in order to pump products. P means product. The difference V by W average energy content of S and that of transition state is called as activation energy. Enzyme eventually bring down this energy barrier, making the transition of substrate to product more easily. That means this indicates what? Enzyme that helps in lowering the activation energy. It helps in lowering the activation energy and getting the transition state by using a little amount of activation energy. So this graph that explains uh, difference between PW average energy content of substrate and that of transition state is called as activation energy, and enzymes usually uh, they helps in lowering the activation energy. Then here is a slide that is mechanism of enzymes on substrates. And this mechanism is of two types. One is lock and key hypothesis, and the other one is induced fit theory. First one, what is that? Lock and key hypothesis. And this was discovered by Emil Fischer, scientist. And here, enzyme molecule and the substrate molecule both will here substrate molecule, this is substrate and the a lock and the key model, lock and key model. Here, this is the substrate molecule and this one key is enzyme molecule. Here, substrate molecule and this is enzyme. Now, This one is enzyme and this key is substrate molecule. Substrate and a key. Here, enzyme molecule is there and these are the two substrate molecules. Now, this enzyme is having active sites in different shapes. Enzyme molecule, it is having different shapes of active sites. And according to this, substrate molecules are also there. See here, one is round in shape and the other one is in the shape of W. Okay. Now, during the reaction, this enzyme active sites, they are filled with they will be filled with substrate molecules. Say this one. And this substrate molecule, according to its shape, it comes and fit in this active site, and this one, this substrate molecule, according to its shape, it comes and fit in this active sites. By that, say, here a temporary bond was laid between the substrate molecule and enzyme. Here also, substrate molecule and enzymes. Both the molecules, substrate molecules, fit in their active sites according to their shape or shape of the active sites on the enzymes. By this arrangement, there becomes a complex which is called as enzyme substrate complex. And after this enzyme substrate complex stage, it is temporary. It is not permanent. It is temporary. After some time, furtherly, it results in the formation of end products. And a new bond will be laid between the two substrate molecules in giving this product, end product. And this enzyme that comes out as it is with the same shape of the active site. Look at the diagrams here. Starting diagram. This is enzyme with active sites, and the same enzyme is coming out after the reaction. At the starting of the reaction, enzyme is in this way, in this shape. And after completing the reaction, the enzyme is as it is without undergoing any change. So, according to the definition, enzymes are the biochemical substances which participates in the reaction to enhance the rate of reaction without the participation in reaction. Okay, so according to the definition, it is proved that enzyme is not undergoing any change, and that this substrate molecules, by making a new bond between them, they are coming out the form of end products, isn't it? So this is the lock and key theory. That means here in our daily life, we closes the door by arranging a lock and that means a lock is closed by using its a concerned key only. That means that key should have the same shape of this lock and then only this key will be inserted in the lock and the lock is closed. And the same way, this enzyme with having with active site should resemble the key. Then only this key that will be fitted or that will be set in this active sites, and a bond will be laid between the lock and the key. And then only this lock opens and the key comes out as it is without undergoing any change. The same is proved in this enzymes activity or enzymes mechanism. According to the shape of the active sites, substrate molecules they comes and fit in this active sites, and as a result of that, a complex will be produced, which is temporary. And after the formation of enzyme substrate complex, it stays for some time here in this stage, and furtherly that undergoes a reaction and releases end product by giving a new bond between the substrate molecules and the enzyme that comes out as it is. So this is one type of hypothesis which is laid by Emil Fischer. The next is and the next induced fit theory. Next hypothesis is under the mechanism of enzymes. Next hypothesis is induced fit theory. According to this, shape of the substrate molecules, shape of the substrate molecules, they resembles the shape of the active sites. And here, substrate molecule that comes and fit in this active site, and this one also comes in this site, and then a new bond will be laid between the two substrate molecules, and that new end product that comes out by leaving this enzyme as it is. But here in this induced fit theory, initially this hypothesis was discovered by Koshland, and here in this induced fit theory, enzyme has two active sites. Here in this diagram, it was not mentioned. Active sites are of two types. One is one is one is one is catalyzing site, and the other one is buttressing site. This catalyzing site, it has in configurational change and creating new bonds or breaking the bonds between the substrate molecules. And the other one is buttressing site. This buttressing site helps in attaching the substrate molecules. So in this induced fit theory, each and every enzyme has two sites. One is catalytic site, the other one is buttressing site. Buttressing site that helps in attaching the substrate molecules, and catalyzing or catalytic site that helps in breaking the bonds or making the new bonds between the substrate molecules. So according to this, after the attachment of substrate molecules to the buttressing site, this catalytic site that bends towards the substrate molecules. That means it comes very closer to the substrate molecules, and this catalytic site that helps in creating new bonds between the substrate molecules, and it helps in breaking the bonds between the substrate molecules. So the catalyzing site, it helps in creating new bonds or breaking the bonds. So according to the situation, it helps in creating new end products. And after that enzyme, it comes out as easily an end product that will be released. And here, this buttressing site, it is not the site for attaching with the end products. It is a site for only receiving the substrate molecules. That is most important thing. So in induced fit theory, this buttressing site helps in only attaching with the substrate molecules, but not with the end product molecules. So here, this induced fit theory, which was introduced by Koshland, explains what it explains, or what he explained was each and every enzyme has two sites, and one site is called as catalytic site, the other one is called as buttressing site. And buttressing site, it helps in attaching the substrate molecules, and catalytic side, it helps in the configurational change. Uh, it shows by showing configurational change. It helps in breaking or making the bonds between the substrate molecules. As per that theory only, here we are observing a complex, same in that of lock and key theory. Enzyme and substrate molecules both togetherly makes or frames a complex which is called as enzyme substrate complex. And this is a temporary state. And furtherly, the substrate dissociates into end product and an enzyme as it easily. So this enzymes, they are influenced by various factors like temperature and yes. Here, classification of enzymes. Classification of enzymes. Basically, all the enzymes are proteinaceous in nature, and this proteinaceous enzymes, they are classified into simple enzymes and conjugative enzymes. Simple means they are made with only enzymes. That means proteins only. Only protein part takes part in the synthesis of enzymes. That's why they are called as simple enzymes. The next one is conjugate enzymes. That means these enzymes, they are made with protein part and non-protein part also. And that type of enzymes are called as conjugate enzymes. And in this, protein part is called as apoenzyme, non-protein is called as cofactor. And here, these cofactors may be metallic or organic. They may be different types. Here, different types of cofactors. Cofactors may be either metal ions, non-protein parts. Cofactors means non-protein parts. They may be either metal ions or organic cofactors. And these metal ions, they are tightly attached with the protein part. And organic cofactors, they are loosely attached with the protein part. So here, metal ions, when they are cofactors, they attaches the protein part tightly and they are not easily detachable. And organic cofactors, they are loosely attached with the protein part and they can easily detachable from the protein part. The next, organic cofactors. If the coenzymes, they are loosely associated with the apoenzyme, example, NAD, nicotinic acid, and CoA, pantothenic acid. And prosthetic group, if it is tightly bonded to the apoenzyme, example, heme group of the enzyme peroxidase. Then classification and nomenclature of enzymes. Classification and nomenclature of enzymes. And here, according to IUB, International Union of Biochemistry, which was established in 1964, classified the enzymes into 6 major groups, and they are oxidoreductases or dehydrogenases. Then second one is transferases, and third is hydrolases, and fourth is lyases, and fifth is isomerases. Fifth is isomerases, and sixth one is ligases. These are the important main groups. Important main groups are major groups, and this was made by IUB, International Union of Biochemistry.